We have generated massive amounts of genomic data over the last 2 decades. If you try to trace the regulatory grammar that builds a plant from a single cell, you hit a wall. Our data is highly fragmented. We have built incredibly detailed islands of knowledge around a tiny fraction of species, specifically laboratory model organisms like Arabidopsis and major staple crops like corn and rice. Outside these heavily sequenced clusters lies a vast, uncharacterized phylogenetic ocean. We possess immense molecular detail on a few highly domesticated plants, but almost nothing connecting them. This sampling bias leaves us blind to the macroevolutionary logic that allows wild species to survive extreme heat, drought, and pathogens. Without mapping the genomic steps that connect wild adaptations to domesticated biology, we cannot easily transfer those survival traits to our crops. This maps 64 angiosperm orders. Gray pie charts show a massive blind spot. 95% of plant genera and 51% of orders lack a reference genome. Unsequenced basal lineages like ceratophyllales and volleyales obscure early flowering plant divergence and complex trait evolution. To map this missing architecture, researchers have launched the PLANeT initiative. This international consortium is working to sequence the entire plant tree of life, capturing the full spectrum of genomic diversity across 470 million years of evolution. Instead of maintaining isolated data points, PLANeT will compile a single, standardized digital library of natural problem-solving. Modern agricultural breeding faces a structural limitation. Successive domestication bottlenecks have severely reduced the genetic diversity of our crop species, which steadily increases their burden of deleterious mutations, known as the genetic load. To identify these traits, geneticists rely on tools like genome-wide association studies, or GWAS. This acts as a high-resolution microscope, scanning a crop's genome to find statistical links between a DNA variant and a physical trait like yield. But this microscope is inherently noisy. Because large blocks of DNA are inherited together, a property called linkage disequilibrium, GWAS regularly flags hundreds of candidate mutations simultaneously. Most of these are neutral passengers. This data visualization shows a typical GWAS Manhattan plot. The high-resolution data alone cannot mathematically distinguish the single true causal mutation from the hundreds of tightly clustered neighboring variants. High-density data is insufficient on its own. Researchers require a secondary lens to filter out the noise accumulated during the short window of human agriculture. That secondary lens is the phylogenomic telescope. By comparing sequences across distantly related taxa, from ancient non-flowering lineages up to modern eudicots, we can observe natural experimentation across millions of years. This deep time view exposes the mechanics of purifying selection. If a specific nucleotide is universally critical for a plant's survival, it is forbidden from mutating. We locate these critical sites by aligning the genome sequences of wildly different species. If a nucleotide remains identical across extensive evolutionary divergence, it identifies a highly constrained, functional sequence. When we apply this deep time constraint map directly over our GWAS data, the neutral passenger mutations are filtered out. We are left with the exact constrained variants driving the target trait. Identifying these constrained sites gives geneticists the exact coordinates needed to engineer crops with high precision, targeting mutations that either enhance fitness or violate 470 million years of evolutionary rules. This phylogenetic diversity serves an even larger computational purpose. It provides the strict training infrastructure required for DNA-based artificial intelligence. AI foundation models scale mathematically with sequence variants. Models trained exclusively on the narrow genetic base of modern crops lack the diversity to learn robust regulatory parameters. They require the full breadth of the plant kingdom to calculate accurate weights. Fed with diverse inputs, these models execute multi-level analysis, starting with structural annotation of non-coding sequences, mapping complex protein interactions, and eventually discovering novel gene modules for high-value traits. A completed phylogenetic map enables zero-shot inference, granting researchers the ability to predict the function and biological effect of any genetic variant directly from the sequence, bypassing years of laboratory testing. This computational framework directly accelerates natural product discovery. Wild plants produce highly complex chemical compounds with rigid, three-dimensional structures that are extraordinarily difficult to synthesize chemically in a lab. Complex survival mechanisms, like carnivory, mycoheterotrophy, or the production of targeted medicinal compounds, have evolved multiple times across completely distinct branches of the plant family tree. This phylogenetic tree maps the distribution of heterotrophic traits across diverse angiosperms. By analyzing how these traits arose convergently, AI can scan the genome to identify the shared regulatory grammar and locate the exact biosynthetic gene clusters responsible for chemical production. The PLANeT data provides the genomic blueprints needed to reverse engineer these pathways. We can extract the sequence, insert it into cellular manufacturing platforms, and metabolically produce therapeutics that were previously inaccessible in wild species. To build this infrastructure, the PLANeT Consortium is coordinating over 100 laboratories worldwide to train 1,000 scientists, standardizing genomic outputs to eliminate analytical redundancy. The result is a 470 million-year digital library. This database supplies the specific mathematical tools required to assess the adaptive potential of endangered species and engineer localized climate resilience into the global food supply. Within decades, field biologists will carry portable sequencing arrays, instantly analyzing DNA on site to read the biological mysteries encoded inside any species on Earth. By illuminating the complete evolutionary history of land plants, we move from reactive conservation to precise biological design, securing a resilient and sustainable future.